Well, hello everyone. My name is Dawn Van Dam, and I am the president of 1 Million Solutions in Health. So welcome to today's Signature Square presentation. We're really thrilled to be bringing you this opportunity to take a look at some fabulous new technology. And what we ask of you in this format of the Signature Square is we do ask you to give this scientist some specific feedback through the completion of a questionnaire. So we're going to stick that questionnaire right into the chat box in just a minute and ask you to take a look at it as we go through the presentation today. But just to give you a little bit of background as we get started here, I'm going to go through a welcome and introductions. Dr. Kaori Ishida Stansberry is going to do her presentation of gel cell. And then we're going to ask you, as I said, to do your evaluation as you go along. There'll be some closing remarks. I'm hoping that you have lots of questions for our presenter today, and we really would love it if you could complete the survey before you end the session today or shortly thereafter. So I'm looking at a lot of people streaming in, so that's great, terrific. So a little bit about us, 1 Million Solutions in Health. Fundamentally, we're trying to improve healthcare one solution at a time, but do that in some new disruptive ways that have never been done before in order to accelerate the movement of science forward away from the scientist and to the patient. Not that we want it away from you, but we want it to be utilized by the patient and helping people get more cures and success in the market. So we're very excited to be bringing this new technology today. So how do we do that? How do we move science forward? We help connect people, we help people learn and share, we want to present lots of new innovative things all around the world, and we do some of that work through our Technology Evaluation Consortium. So the Signature Square is actually part of the Technology Evaluation Consortium, and I'm going to tell you a little bit about that very quickly before we get things underway. So today's presenter, I'm going to give you a summary of what we'll be hearing. Gel cell is a cell culture system that can mimic the micro-scale physiological conditions of the body. Scientists work with model systems today that are meant to be small-scale versions of real-life conditions in the body. However, many of the scientists work on the micro-level models with cells using plastic-based culture containers that are far from the actual environment of the human body. Since plastic culture containers are more rigid than the normal physiological conditions of the body, the results gained using them cannot be easily translated to the human body. To solve this problem, Dr. Ishida Stansberry developed a cell culture system with a tunable substrate that allows the scientists to replicate the mechanics of physiological, normal, or pathologic conditions. So very interesting what we're going to hear today. So I see a number of people joining in. If you wouldn't mind just telling me where you're from. I'd love to share where people are from. If you can just go to the chat box at the bottom and find my name, Dawn Van Dam, then you can send me a chat message and tell me where you're located. I'd love to share where everybody's located. And that way you'll also get used to using the chat box feature, and during the presentation you can send me any questions that you might have. So I'll wait for you all to tell me where you're from, and I will tell you all about that in a moment. So the Technology Evaluation Consortium. It is a collaborative partnership within the pharmaceutical and biotech industry and other stakeholders. And fundamentally what we are trying to do is be the place for the industry to conduct their evaluations and their validation. So today it's an evaluation, and our overall mission and goal is to triage and streamline new technology consideration for the industry. So I see some people have told me where they're from. I see Greater Boston area. I see India. I see Sao Paulo, Brazil. I see Oxford, the United Kingdom. Ghana. I see Germany. I see Saskatoon, Canada. I see Connecticut. I see Egypt, Italy, Nigeria, Romania, Serbia, Philippines, Canada, Portugal, and quite a few more from the United States. So great, thank you for that information. Great to see we have a global, we have a Chicago as well. I forgot that one. Great to see our people from all around the world joining us today. Thank you. So as I said, the Technology Evaluation Consortium, on the one side we have potential end users in the pharmaceutical and biotech industry, and on the other side we have technology vendors or scientists sharing their science. We put them together, we talk to the scientists beforehand, make sure their presentation is prime time ready, so to speak, and then get them to share it with you, and we rely on you to give us great feedback. Here are some of the people who have been involved in the Technology Evaluation Consortium, either over the years or currently. I can see one person on this slide who's actually in attendance today. I won't name them, just not to call them out. And then here's another slide of other people who have been involved in the Technology Evaluation Consortium either today or over the years. So some great names around the industry, and we're really pleased to have everybody joining in. So our process is simple overall. We find new scientific solutions. We gather input to optimize the technology either through programs like the Signature Square, which we're doing today, or our TEC validation projects, where we actually get the scientists to roll up their sleeves in the industry and try out your new science. So that's quite exciting project. And then we share the findings with our community and our consortium members. So today's Signature Square, I gave you a little overview of the abstract, but we're going to have the science presented, and you're going to give us feedback. And we've already put the survey into the chat box, so if you want to open it up and start to get familiar with it, fill in any of the information, that would be great. And really, it's so very important for this particular scientist and for all scientists to get this information. We do provide them with a summary report at the end of both your quantitative and your qualitative feedback. Nothing will be attributed to you specifically or your company or anything like that, but it is a fabulous way to accelerate their perfection of what they're offering to the market and get them to have feedback that's so important for their success. We rate them in the end on one of 4 areas, whether they're an upstart, a blockbuster, an innovator, or a novel. And that comes from the numerical rating. The opportunity for the industry is for them to learn about new ways to enhance the productivity of the research and development process, whether with new safety tools or new research tools, new discovery tools, development tools, whatever might be the case, and to then therefore make sure that we get these great items in the area of science out there to the market faster and quicker. So I already gave you the overview of the presentation. Very excited to have this opportunity today. Just wanted to highlight a few more things. Make sure, see what I haven't covered here. So gel cell can replace cell culture flasks without any change to current protocols for the cell culture and will provide results that are comparable to physiological conditions. This innovative technology will also lead to a decrease in the need for animal models. It saves time versus hydrogel-based cell culture because of the significant increase, at least 4 times higher, in the surface area for cell harvest. And most importantly, it will provide for the culture of primary cells acquired from any tissue of the body, as well as for stem cells. So we have lots of different usage areas and information about this today. So let me tell you about our speaker, and I'll jump to her biography here real quick. So Dr. Kaori Ishida Stansbury received her Bachelor of Pharmacy degree from the School of Pharmaceutical Sciences at Nagasaki University and her PhD degree in Biomedical Sciences from the Graduate School of Medicine at Kagoshima University, Japan, with a focus on cell and development biology. Dr. Ishida Stansbury's postdoctoral work was conducted at the prestigious laboratory of Nobel laureate Dr. George Palade, at the University of California in San Diego, with a focus on vascular biology, and at the Cardiovascular and Pulmonary Research Group at the University of Colorado Health Sciences Center, with a focus on vascular cell biology and pulmonary arterial hypertension. Dr. Ishida Stansbury was later recruited as research associate and quickly promoted to a research assistant professor in the Department of Pathology and Laboratory Medicine at the University of Pennsylvania in Philadelphia. Recently, Dr. Kaori Ishida Stansbury's studies have focused on the role of microenvironment in vascular cell growth and functional maturation in embryo and pathological conditions, which include the utilization of biomimetic substrates. Dr. Ishita Sandberry is currently a Research Assistant Professor and Associate Director, PHBI Penn Cell Center in the Department of Pathology and Laboratory Medicine at the University of Pennsylvania. So with no further ado, I think my next job, yes, is to remind you to fill out the survey and also to pass over the baton to Dr. Ishita Sandberry for her to give you her presentation. We just, yep, there you go.
Hello?
Yep, we can hear you perfectly. Over to you.
Thank you. So thank you for the kind introduction and giving me the opportunity to present gel cell device. Most of the advancements in science and medicine have been originated using cell culture as a small convenient model system. Scientists have been isolating cells from healthy or diseased organs and growing cells to test potential therapy for centuries. However, the cell culture technology has not changed much. The traditional cell culture system relies on plastic culture dishes with cell grown on the rigid surface, that is the far side of the plastic plate. Major problem that has been largely ignored in this classic culture model is stiffness of growth surface. Each organ in the body has different stiffness from very soft brain tissue to hard bone tissue. Convenient plastic culture dishes are extremely hard surface and cannot provide stiffness in the body where cells are originated. So recent studies have shown that surface stiffness greatly controls cell development. It is time to bring cell culture systems that much closer to real-life conditions in the body. So gel cell is a tunable cell culture system that can be customized to a specific stiffness and size of surface area from cover glass size to the larger glass plate size. With the culture cassette, it is easier to seed and culture the cells on the soft gel. Gel cell technology relies on hydrogels that are fabricated on top of glass. So for instance, if you want to grow brain cell on soft environment similar to that in the body, you can now conveniently grow cells using gel cell. Advantages of gel cell technology are numerous, including the realistic environment that is similar to the body, affordable price comparing to other chemical origin expensive hydrogels, reduced reliance on animal models, and reduce time and steps for translation into therapies and drugs. Here's the model of how we make the cassette and the cell culture surface. Particularly gel cells unique feature large surface culture area with preferable stiffness make it easier to expand stem cell or other type of cell number with maintaining their characteristics and easily harvest materials for future research from basic research to drug discovery and cell therapies. In our knowledge, gel cell is the first culture system that overcomes the previous problem that limited to cover glass size. So primary prototype has been created and successfully tested in our lab. Using this gel cell system, we have tested and published the growth of embryonic lung vascular cells, which you can find on our publication. As the slide shows, the stiffness changed the protein expression from top to bottom because the top two panels are 500 pascals, bottom is about 3 times stiffer, and you can see the color is translated to protein expression, and this green or red fluorescent protein expression changed by the stiffness, and also change the shape, and effect of growth factor also changed by stiffness. By using large surface gel, we could easily harvest RNA material to examine the gene expression. Here, the right side, we demonstrate that smooth muscle maturation marker gene expression is stiffness dependent. This could not be easily harvested if you need to harvest same quantity of cell materials from multiple cover glass size gels. Next is another example of using gel cell. This is our ongoing research to find the cause of pulmonary arterial hypertension. Pulmonary hypertension is a rare but lethal disease that occurs in the arteries of the lungs. The arteries of the patient lungs have high blood pressure with abnormally thickening and stiffened vessel wall that eventually leads to heart failure to death. Pathological studies have shown that the cells on the vessel wall abnormally grow, which contribute to the wall thickening. However, cause of this cell behavior is unknown and that makes it difficult to develop targeted therapies. To begin to understand pulmonary arterial hypertension, first we examine the normal vascular cell behavior on different stiffness substrate using mouse origin vascular cells. Image shows that the mouse origin lung vascular cells seated on different stiffness and cultures for 24 hours and the 48 hours. The cell did respond to the stiffness. Cells on the soft 400 pascal substrate tend to be more square shaped with less cell growth between 24 hours to 48 hours. However, as the substrate stiffness rises, cells are more elongated and the numerous. You can see the compare 400 pascal and 12 kilopascal. We quantify the cell growth ratio and shape changes. As you can see, cell growth ratio on the left side and shape change that we translate by aspect ratio of the cell are depending on the substrate stiffness, and the 12 kilopascal is still the softer than the rigid glass or plastic plate, and they depend on the stiffness rises, then cell growth is increased. Cell morphology also changed by the stiffness. And results from mimicking the physiological range of stiffness that is in the lung between 3,000 to 5,000 pascal is different from the cells on plastic or glass that is on the far right side of the graph. Next, we tested human pulmonary artery smooth muscle cells from control subjects and cultured on the different stiffness of substrate. Images show the cells seated on the different stiffnesses and cultured for 24 and 48 hours. Once again, the cells did respond to the stiffness as mouse cell dead. Cells on 400 Pascal substrate tend to be more square-shaped with minimum cell growth. And as the substrate stiffness rises, cells become significantly more elongated and numerous. Quantification confirms that both mouse and human control cell growth respond to the stiffness in the same manner. Finally, we tested disease subject origin cells on the substrate, which showed different response. Here I show all the graphs side by side from PAC1 means the mouse origin pulmonary artery cell, and the middle normal NHPS smooth muscle cell means the control human pulmonary artery smooth muscle cell, and far right is the patient's pulmonary arterial hypertension cell. We have found that the patient's smooth muscle cell had a different response to the stiffness compared to the mouse and human control smooth muscle cells. Patient cells seem to lose the sense to stiffness and grow constantly higher rate. These results demonstrate that advantage of gel cell system not only detect fundamental discovery, but also developing efficient therapies because the results using this system closely translate into the in vivo condition. Therefore, researchers can reduce the costly animal use and reduce the multiple experiments using conventional culture system-based experiments. It will be for long-term time and cost effective. Our primary market is current cell-based biology and biomedical research laboratories in both academia and industry. By replacing the existing conventional culture plate, we believe that we can expand the market to industry and academia laboratories studying biotechnology, tissue engineering, stem cell engineering, regenerative medicine, system biology, and the new science field. Current status of the project is at the laboratory prototype level that we have tested with multiple human and animal origin cell lines. We are currently seeking non-dilutive funding from a variety of sources with goal to have industry standard device in the next 6 months. And we need to design and develop an industry standard product ready for product line quality of the culture device with industry partners. And the timeline is first 6 months, we will develop industry standard device models. In parallel, we will initiate the marketing by setting the website and other social communication and the direct contact with possible users for reference. In the following 6 months, we'll use the machine shop in Philadelphia to design and develop a robust outer container for the device. We believe that the 3D cell culture, even 3D cell culture market is valued at $400 million and is expected to grow to about $2.2 billion by 2019. Any lab using cell line and cell culture stands to benefit from switching to gel cell. Thank you for the opportunity again. And if you have any questions, please contact me. Thank you.
Great, thanks so much, Dr. Sheeta Stansbury. And I'm just going to ask for the ball to come back to me. We would like you to set all the people listening in to send in questions now through the chat box. And the opportunity is, again, for you to fill out that survey and give some terrific feedback to Dr. Carrie Sheeta Stansbury. But importantly, one of the questions in there is actually for us to do through our consortium a validation project with this technology. So when you get to that question there, if you think that it would be a good idea to do a validation project with this technology through the consortium, please let us know. That would be really helpful. So I see some questions coming in already, and I'm going to encourage people to send in more. So I'm going to start to ask you some questions, Kiori, and then we'll go from there. So the first question is, what is the major difference using this gel and using conventional culture flasks and dishes?
Yes, as I presented, each organ has different consistency, stiffness in the body. That's why the result using gel cell culture system with physiologically relevant stiffness are much closely translated to the cell response in the body compared to the result using conventional plastic culture flask and plate.
Okay. Sorry, go ahead. What are you going to say?
That is a huge difference because, again, that's the conventional plastic have megapascal really rigid conditions, even harder than bone conditions. So imagine if you are culturing brain cells on that condition, they don't respond, they don't grow, and even grow, the response to some chemicals are totally different from what's happening in the body.
Right, okay. So another question here is, much of the industry-related cell culture is conducted at high throughput. So for instance, 96 or 384 well culture plates. Are there currently gel cell formats that are compatible with these micro well plates, or if not, are there plans? Like, so where are you at right now?
We haven't tested in smaller surface, but the smaller surface like the 96 well, but as small as 18 millimeters glass, cover glass size, we could create the gel. So going to smaller size, we don't see any problem.
Okay. And so from a production perspective, are you ready to do that now, or do you need more time?
So again, current condition is laboratory prototype. So we need to go production line. We need to develop the cosmetic, and we need to develop better industrial standard material.
Right. Okay. Okay. So there's an area where you need some industry advice and support. So, and if there's a follow-up question to that, just stick it in the chat box, please. Is there any advantage using this product for laboratories that use animal models? Would this product result in any help or savings for these labs?
I believe that large reduction in live animal testing would be realized because the gel would give much more accurate results that in reducing the need to live animal to quite standardize the results, and perhaps as high as the probably this gel has the potential to save labs that maintain live animal facilities a great deal of money, both the short and long term, because from so far researchers plan the experiment from digit plate results to jump into the animal, so you cannot pinpoint the in vivo condition, but using gel cell, it simulates the body conditions, so you can definitely reduce the animal use.
Okay, great, thank you. And I'll just encourage everybody to send in any more questions that they have. But here's another question for you. What kind of cells have been tested on this device? So besides maybe the ones you mentioned, or just to recap.
So in our lab, we have tested embryonic precursor cells, which developed to vascular cells. And we also tested, again, I presented that tested human adult lung vascular cells from control and patients. And our collaborator also have tested the cells from liver and mesenchymal stem cell, and they also have interesting results responding to sickness.
Okay, thank you. Great. So another question here is, does gel cell culture interfere with microscopy or spectroscopy, colorimetric assays? Is there a colorless version available?
Yes, the pink color is because we hydrate with the phenol-containing media. So by using phenol-less media, you can remove that pink color. And again, our advantage is gels are on top of the glass, not plastic. So the fluorescent reflection is reduced.
Okay, great. Thank you for that clarification. So thinking about the future, potentially what kind of cells could you use with this system, with this device?
Any kind of cell, but I said because it's the hot topic that neuroscience, so neuron cell, brain origin cell, that is difficult to grow in rigid surface. So soft gel have advantage on it, and other application and major reflection to the therapy is stem cell culture.
Okay, terrific, thank you. And again, I'll just remind anyone to send in any other questions. How about your IP status? Can you tell us a little bit about the IP status?
The Innovation Center at UPenn, University of Pennsylvania have been working for protecting this property. The current condition is the gel has not been released to anyone, and that for any further IP questions, if you have any, please contact Vivian Martin at the Innovation Center. If you need her contact information, please, I can send her contact information.
So I'm just going to put up actually my contact information think it's here. Yeah, so people can maybe contact me, and then I can get them in contact with you in the group if they want to do a specific project or anything like that. Okay, let me see what other questions are here. Is there any shelf life? Or what's the shelf life?
Is the current prototype of the gel have short shelf life because once it's hydrogel, so if once hydrated and you have to use. However, there is a potential that we can develop for longer shelf life to several months if refrigerated. That is the current condition.
Okay, great. And I know we're today talking about the usage in, you know, the R&D pathway, but you see usage in many other areas. So can you maybe just give a recap of those other areas that you think it'll have potential usage in?
The usage application would be the neurobiology, stem cell biology, bioengineering, for example, synthetic organ development, and so any field to develop therapy again, and therapy, pharmaceutical companies, the drug development, that is the field to have maximum benefit using this cell.
Okay, great. So I'm just going to remind everyone just to click through on that survey before we end the webinar. The link is right there. And that way, even if you haven't finished it, you can still do it afterwards. If you don't get a chance to click through on the link that's in the chat box, we can always send it to you afterwards. But we would really love you to fill out the survey and give us your feedback. So just before I move to closing remarks, just wondering if anyone has any experience that they want to share to give Dr. Carrie Stansbury any thoughts or advice, any level of interest that they want to share in the chat box, or any additional questions or information that you'd like to have answered. I think I've covered every question that was sent in the chat box so far. I will just double check while I'm waiting to see if anybody else has any other questions. And then, Carrie, I'll turn it to you for some closing summary remarks just before we end off the session today. So let me just give everyone 10 seconds more to send in any questions. And if not, we'll turn it over to you. Carrie, so just any recap that you want to give of your gel cell technology?
Yes, we believe that gel cell technology with tunable cell culture system is the next generation of the culture system, because we are essentially we are using just glass plate, so it's the time to use more efficient the culture system and to replace the conventional plate culture system. So this I believe this one is the next generation, and please, any feedback is appreciated.
Great. Well, thank you so much for taking the time to share your discoveries and your work with us. And I'm sure the audience will have lots of follow-up questions. And again, just to remind everyone to please go in and fill out the survey. It should only take you a few minutes, but we would like some qualitative feedback as well, if you wouldn't mind. That would be fabulous. So again, thank you very much to Dr. Carrie Stansbury and to all the participants today from around the world. We appreciate you either staying up late or getting up early, whichever might be the case. And we look forward to bringing you lots of great science in the near future. Have a great day, everyone, and have a terrific week. Bye now.
Thank you.
Thanks.